And with that, I want to introduce today's speaker and her presentation. Kristen, you can go ahead and share your screen. We're really excited about hearing some of this really hot off the press results. So Dr. Kristen Behrens has been with us here in Ann Arbor since about May of 2025, so not quite a year. She's a postdoctoral research fellow working. She's employed by the Cooperative Institute for Great Lakes Research, so SIGLR, which is affiliated with the University of Michigan. But she also is supervised by Amanda Ackus, or mentored by Amanda Ackus here at the USGS Great Lakes Science Center, and that's the lab where she physically does her day-to-day work. Her expertise is using genomics to address questions regarding speciation and sex determination in fishes. She received her BS and master's degree in BS in biology and her master's in bioinformatics, both of those at Juniata College. Is that right, Kristen?
Yep.
Yeah. Where is Juniata College?
Central Pennsylvania, just below Penn State.
Oh, very good. Okay. And then she went on to the University of Maryland, uh, where she got her PhD in biological sciences. Her research there focused on cichlid fish sex chromosomes. Today, she's going to be talking to us about a new system. Oh, there you go, Marissa, another Unionic grad. She's going to be talking to us about a new system she's been learning, new species complex, genomic insights into the population structure and sex determination in Great Lakes ciscoes. And she said that she's really excited for you to ask her some really in-depth ecological questions about ciscoes. So make sure you, I'm just kidding. No, we'll work those out. But Kristen, her webinar is about 40 minutes or so, and she has graciously agreed to stick around for 15 to 20 minutes or so of questions and discussion. So take it away. And thanks again for volunteering to do this.
Awesome. Thanks, Bo. Yeah, so like Bo said, I'm going to be turfing a genetics talk at you guys today. So I'm going to do my best to keep you with me. So this talk is kind of divided into two parts. So I'll talk first about lake stuff at the population level, and then I'll swerve us around and talk about sex, which is where a lot of my previous work has been. So this isn't really a group that needs a lot of introduction to chorogonines or ciscoes, but I will rehash some of the many problems that are associated with working with this group. So obviously the first one is that these fish are very morphologically similar to each other. We have a really hard time distinguishing them based on physical characteristics. And another problem we face is we don't really understand the divergence between these species. We don't understand where they fit phylogenetically and how they broke out into these different things and how they did that, what underlying traits are driving speciation. We also know that there is some level of hybridization going on between some of these species, particularly between Chorogonus ardei and hoyi. However, we don't really understand the extent of that hybridization at this point because we haven't addressed it really from a genetic standpoint. We don't really understand either the underlying population genetics within and between the lakes. So we can think about a species in a lake as a single unit. However, there can be underlying structure that's maybe divided out by location around the lake or other factors that could be important for conservation. So we want to understand whether or not it's a homogeneous population or not. So whether or not there's some of the substructure. And then finally, we would like to understand what the sex determination system or systems in these fish are also for conservation purposes. So I'm sure at this point you're pretty familiar with the conservation framework for restoring chorogonians in the Great Lakes. So we're over on the right side here, starting to use some of these genetic tools to start planning how we could potentially restore these fish to the Great Lakes. And the way that we're going to do this is we're going to take a genetic approach. So we can't tell them apart morphologically. we need to look at what distinguishes these fish genetically. So previous work has started at the microsatellite level. So these tiny little repeats, um, so a small region of the genome to compare between species was how we first tried to distinguish ciscoes from each other. And you can tell from the way all of these colored dots like to pile up on each other that didn't do a really great job of doing that. We can't pull apart what we know are distinct species. And we use a plot that you're going to see a lot today called a principal component analysis plot, or a PCA, where we're pulling out some of this variance onto two different axes that explain a certain amount of the variance. So what we would expect to see is if things are very different, they're going to pull out into separate clusters. And when we started using a newer technology, RADseq, which samples about 1% of the genome, sites around to compare, we start to see Artidae, Hoyi, and Kai start to pull out into these groups on the first and second axes of this plot. And then we can start to, from a genetic perspective, determine how similar these species are. And what we're doing is by increasing the amount of data that we're looking at. So we went from looking at one small region in the genome to maybe 1% of the genome. The more data we have, the better resolution we're able to get. So it's kind of like increasing the pixelation on an image. So we started out with blurry MONA over here, working with microsatellite and RADseq data. But as we do whole genome sequencing, we can start to really resolve the relationships between these fish because they're very similar. And by not capturing the entire genome, we're not capturing the sites that are actually different. So that's what we're going to be doing moving forward. So we have over 1,000 samples from across every lake except, of course, Erie, where there are no longer any coregonines. And what we did is a methodology called low coverage whole genome sequencing, which I'm going to explain in a minute. But we have a really good sampling of contemporary populations from across the lake with which to start to answer the questions that I highlighted at the beginning. So I'm not going to go super in depth on the bioinformatics here. I will spare you that. But basically, we start with a tissue sample. Oh, I am in the dark now. It's fine. A tissue sample, either a fin clip or in some cases a larval fish, because they pop really nicely into a little tube. And then we get DNA from those individuals and we send them out for sequencing. What we get back is just gigabytes upon gigabytes of data. And again, I'll spare you the back end stuff, but essentially what we want to do is called genotype likelihood. So we want to know what the variable sites in the genome are. So these are the sites that are making these fish different from each other. And from there, we can use that data to compare species or sexes or populations. So not as scary, maybe, as you might think. So I will walk through the first part, which are the lake level population results. And I have these broken out by lake and then finally at the end, the riverine populations. So we'll start with a nice and clean one, Lake Ontario. So we have data from sites up at this top right corner of the lake where there are existing populations of coregonines. And again, we're back to our PCAs. You're going to see one of these each, at least for each lake. So we're back to our principal component analyses, and we can see that this spray of colored dots are splitting across two of the axes. So we have two distinguishable populations here. We have a population that's in Big Bay, Bay of Quinte, toward the left side here, and then a population that's in Chameau Bay. So it seems like there are two distinct populations of Coregonus artidae in Lake Ontario, which fortunately coincides with some of the management decisions that have been made recently for managing this population, just based on some very preliminary genetic data. So these two distinct populations might infer efforts to conserve these two, because it's unlikely that one or the other would repopulate the opposing bay if necessary. So this is the other kind of plot you're going to see a lot from me today. We call this a Manhattan plot. So your x-axis here is chromosomal number. You'll notice that 5b and 6b are a little out of order toward the end there. That's because of our fussiness. Your y-axis here is a measure called FST. So this is a measure of differentiation between populations, and we calculate this in a pairwise manner. So our first plot is comparing the population from Big Bay to Shimo Bay. And then we have Big Bay compared to Mexico Bay, and then finally Shimo Bay compared to Mexico Bay. And so what we're interested in are these little outlying regions where divergence between these two populations is elevated, because that might suggest something in that region is involved in these two populations being diverged from each other. And I'm also going to preface and say that all of this data is very preliminary. I was scrambling toward the end of December to get stuff pulled together for this, so we have not yet investigated what's in these regions. That's something that we will be doing moving forward to try and target possibly genes that are involved in these species, populations, et cetera, diverging from each other. So here in Lake Ontario, there are a couple of different regions of interest that we'd like to pursue. Moving on to Lake Michigan, so we have two kind of areas where we sampled. Most of the Ardii is up toward Grand Traverse, and then the Hoyi is more out toward Open Lake. And what you can see on this PCA is that Ardii and Hoyi are pretty distinct from each other, but we have this cloud here in the middle. These are putative, likely F1 hybrids. So we can see in this structure plot here, you get the Coregonus ardii in red and Hoyi in this teal color, and then these hybrids are represented by the teal invading into the red. So we have a handful of potential hybrids between these two species within Lake Michigan. And it seems like Hoyi has a much bigger spread of diversity. So the more this stretches out on the axis, implies within group variants. So more FSTs, and what's really interesting about this comparison of Lake Michigan's Ardii to Hoyi is that they actually have a much higher background FST than we generally see for ciscoes. So this is not an easy plot to pick out regions of interest from, just because there's that much background variation. But generally, a lot of comparisons, you'll see the axes will be much smaller. For Lake Huron, we're seeing some really interesting stuff going on. So this is Coregonus ardii first, and then I'll talk about Hoyi. So we have a lot of spread of sampling, both up toward the North Channel here, as well as Perry Sound and in the Bruce Peninsula area. And what we see on the plot here that I've circled is that we have at least two distinct populations, right, on these first two axes of variation. So Scammon Cove is coming out here in teal, is kind of its own thing. And then Whitney Bay is a secondary population. And what's really interesting is you can see up in the top left of my little map, Whitney Bay and Scammon Cove are practically on top of each other. So it's very interesting to us that despite these little areas being so close to each other, they've formed very distinct populations. And so the second thing we looked at is we can look at other axes of variation. So if we start to look at the third axis, we can see that not only are there distinct populations for Whitney Bay and Scammon Cove, we also get this little group down here that are from Perry Sound. So some other preliminary work had showed that Perry Sound might be different from the rest of the Lake Huron ardii, which we were able to confirm with this increased amount of data. And then finally, everything else in the lake kind of clusters together. Coregonus Hoyi and Lake Huron is a completely different story. So unlike most of the other plots I've shown you so far, there's no real substructure based on group in this plot. So it seems like unlike the ardii in Lake Huron, they haven't really segregated out into specific location-wise populations, or if they had that diversity has been lost over time, which is very interesting to consider. We then compare directly Artidae to Hoyi, and what you can see is, again, they're pretty cleanly separating. You might be tempted to think that these 5 here are hybrids, because that's what they were when we looked at Lake Michigan. However, those are actually the Parry Sound individuals that are just so different from everything else Artidae and Lake Huron that they pull apart from the main cluster, but they are not hybrids with Hoyi. So we went and looked at some of the most interesting comparisons within Huron in a pairwise manner. So of course, Whitney Bay and Scammon Cove are oddballs that are so different, but despite being so close. And there's a region Huron linkage group 37 that I'm probably going to dig into. And then Parry Sound compared to Southampton, just because Parry Sound is so different. And you can see, much like when I compared species within Lake Michigan, there's a lot of background noise. So it's not just a single region or 2 or 3 regions that are different between Parry Sound and Southampton. It's most of the genome. So these fish, these populations have diverged quite significantly from each other. And I want to point out too that the scale on these plots are very different. So here we're only going up to about 0.2, and that's so that you can see these little peaks. However, this plot over here goes up to the maximum FST, which is 1. So there really is a lot of additional noise going on, brought in by the variation found from Parry Sound. So finally, the most challenging lake, I think, to deal with is Lake Superior. So we'll start with Artidae and Lake Superior, and what we find and what we've been finding with GT-seq is that there's really 2 distinct populations of Artidae that we know of at this point. So there is a lineage that I'm going to loosely call Apostle Islands for now, because that's where that group of samples originates from. And then we have this alternate lineage that started showing up around Grand Portage, up here toward the top. But we're really not sure at this point what the origin of these fishes. That's something we'd really like to understand, because they're very, very distinct from the main basin population of Artidae, and they also mess up our analyses by being so different. We also did a comparison of everything we have from Lake Superior. So again, preliminary evidence had suggested the presence of Rhyherdae or shortnose, and this really confirms that there is Rhyherdae in Lake Superior, and it's a very distinct population from everything else in the lake. So we get a nice clean pull of each of the 4 species. There's a couple dots where things are misidentified that have to be corrected. But when we look at this at a structure plot, so all of the individuals are on your x-axis, and then we can see that Coregonus artidae has this weird bunch of little bars here. That's the alternative lineage causing problems. But again, we get pretty clean separation between Hoyi, Kaiai, and Rhyherdae. If we leave Grand Portage off, this becomes a much neater graph. So again, some preliminary comparisons that we thought might be interesting was comparing the main 3 that we've really known about in Lake Superior to Rhyherdae. And what I think is really interesting is on chromosome 32 here, there's a peak that it's kind of hard to see, but it's consistent across all 3 comparisons, which suggests that maybe this is something specific to Rhyherdae, or an adaptation to Rhyherdae that differentiates it from the other 3. And then we have another region here on chromosome 4 that I would like to kind of get in and look at a little bit more, because these are clearly different forms, and we'd like to understand what's really driving that from a morphological or physiological standpoint. Finally, I'm going to talk a little bit about riverine stuff. So we have samples from 3 different rivers that empty into the northern channel of Lake Huron, and that's Mississauga, Serpent, and Spanish River. And so these are roughly Artidae, and what we can see, this is not as nicely clean clustering as some of our other plots, but Spanish River really starts to separate out here from the other rivers. And in order to contextualize these river populations with the lake that they are interconnected with, we compared these rivers to the Artidae from Lake Huron. And what you can see here, and I'll highlight it for you, is the riverine species or riverine individuals, populations, pull out separately from everything else in Lake Huron, which is really interesting because it's telling us that these aren't lake fish swimming upriver. And these were spawning individuals, so we know that they were spawning up these rivers when they were caught. So this is a very distinct group of fish. And then we'll see the way that I already described Lake Huron breaking out, and our Perry Sound individuals here in green. But this is a really interesting result to consider. So obviously we immediately wanted to know, why is Spanish River specifically very different from Mississauga, but also what's going on kind of across these lakes. I flipped the rivers on their side a little bit so I could keep them on the page to remind you that geographically Spanish and Mississauga are the most distant from each other, and Serpent kind of sits in the middle. So thankfully, not a lot of background noise in these plots. I'll draw attention to the fact that this Spanish Mississauga comparison has a larger axis than the other two because there was more variation between these two populations. And there are a lot of regions here that I am very excited to kind of get in and look at to see if these fish that swim a lot farther up the Spanish than they're able to get in the other two rivers have something different going on. But even more interesting is, and this is hot off the press data from yesterday, we went and compared all of the rivers as like a single group against Lake Huron Artidae from a pairwise FST standpoint. And what's super cool is we have firstly a region that's consistent between these two populations. So first we compared to Southampton, which is over here on the right side of Huron, and then Whitney Bay, which is up toward the North Channel. So Southampton to these riverine populations is a very distant comparison. There's pretty much no chance that those two groups of fish are interacting. However, when we compare to Whitney Bay, which is up towards that area, we get this really interesting peak on chromosome 8 that's a very solid. You'll notice that the dots practically stack on top of each other rather than kind of spacing out. And when we zoom in and look at this region, it's about a megabase of chromosome 8. So I will be very soon mining in there for potentially interesting things that might have helped these fish adapt to this riverine behavior where they're running the rivers rather than spawning in the lakes. So that'll wrap up the population genetic aspect of this talk. And now I'm going to take a little turn into sex chromosomes. And I'm going to give a little bit of background first, because I know from experience that this is something that is less familiar to most people. So what is a sex chromosome, at least for what we need to know? Most people at this point are familiar and comfortable with the fact that mammals have an XXXY system where females are homogametic and males are heterogametic, or in simpler terms, the male has the chromosome that's different, which is in this case a Y. It gets more complicated than that. So for example, birds have what we call a ZZ-ZW system. So in this case, the female is actually heterogametic and she carries the W. And in most of these situations, we assume that the gene that's driving sex determination, the region, is on either the Y or the W. Our fishes are, of course, much more complex than anything we see going on in birds or mammals, generally speaking. So they can have XXXY systems, they can have ZZZW systems, they can have multiple systems at once, or they can have environmentally determined sex, whether it's by temperature or social cues. So there's a lot of complexity here when we're trying to determine what's causing sex in these individuals. The other complexity is what the chromosomes that are determining sex look like. So mammals and birds, which have really ancient systems, have what we call heteromorphic sex chromosomes, which means that usually the W or the Y is visibly different under the microscope from its pair, the X or the Z. In mammals, that's usually a shrunken Y because it's lost a lot of sequence. Fish are, of course, difficult. They do not usually have visually distinguishable sex chromosomes. So we can't just pop some blood under a microscope and say, okay, those two matching chromosomes, one is a really different size, that's probably the sex chromosome. We're not that lucky with fish. So we have to employ different methods to try and figure out what their sex determining system is. And the other thing is, unlike mammals and birds, fish change what their sex determining system is over the course of evolutionary time. So we have to think about how they get these new systems. And usually our canonical idea of how this happens is that you start out with a pair of autosomes or normal chromosomes, and one of them gets a new sex determining locus. So this could be a gene such as GSDF, which is big in fishes, or something else completely unrelated, takes the role of determining sex. And that makes this now the sex determining sex chromosome pair. And in this case, we'll use an X and a Y example. And you can get kind of expansion of these regions. So usually the thought is that a sexually antagonistic allele. So in this example, the really colorful displays used by male guppies is thought to be a sexually antagonistic trait. The females don't want to be colorful. It makes them an easy snack. However, it's worth it for the males to have this trait in order to court females. So what happens is this trait exists on the Y. It never occurs in the females. The females don't have to worry about getting eaten, but the males are able to employ this as a courtship behavior. So you get a suppression of recombination between these two. So if you remember, the way that we get variation in our genome is that in meiosis, our chromosomes cross over, they trade little bits and tricks of stuff, and that stops in sex determining regions, because what that does is it breaks up this favorable combination. We want those two traits to stay together. And over time, you can get kind of expansion of this region. It doesn't have to expand, but some fish have sex determining regions that make up most of a chromosome. And over time, as we see in the human Y, that region deteriorates. It loses genes, it accumulates deleterious stuff because it's not purging that stuff. And often this is driven by some sort of structural rearrangement. So an inversion where you get to flip the DNA sequence or something along those lines is one way that you can stop this recombination. So I'm not going to spend a whole lot of time on the slide. I just want to highlight that sex chromosomes can look different from each other. I especially want to talk about takafugu. So these cute little pufferfish, their sex determining system is a single base pair change in a single gene, and that is it. It is not expanded from that. The only way they were able to detect this is by doing a bunch of family crosses to determine linkage. But you can also have chromosomes fused together, or you can get these really big regions. So there's a lot of variation in sex chromosomes generally. So when I start to go in and look at chorogonian sex chromosomes, it could be anything essentially. We do know what determines sex in most chorogonids, or sorry, most salmonids at this point, but we do not know what determines sex in chorogonians. So in salmonids, it's a gene called SDY. This was actually, it's a copy of what was once an immune gene, and it took on this new function of determining sex. It's a little extra complicated than some of what I just described to you because this gene can actually move around the genome. It's what we call a jumping gene. It's transposon mediated, so these little repeats move it around the genome. So it's on different chromosomes across some salmonids, but it's the same gene determining sex. So in 2013, Yano et al. Did a quick PCR. So what they're looking at here is they're trying to see, is this present in males and is it present in females? So you can see across all of the males, they all have a band in this location that suggests that that gene is present. However, our Corygonians here have the bands in both males and females. And that is not what you would expect if this gene was determining sex in Corygonians. So we know they have something completely different from whatever's going on in salmonids. And usually, so this is going to be similar here to the Manhattan plots I've shown you. It's just on a log scale. So usually you get nice clean signals of where there might be things involved in sex determination. This is Atlantic salmon. They see signal on chromosome 2 and chromosome 6. Good for them. They can move on with their lives and start trying to figure out what gene or genes is involved there. We are not so lucky in Corygonians, at least not so far. So I'm going to go through all of the species that we've looked at, and it's not going to be very exciting. I'm going to tell you that right now. So you'll notice these axes are really small to start with. This one's up to 0.3, but there's really not much going on. There's no strong signal for anything in Ontario Artidae. Maybe a little tiny bit in Michigan Artidae and a little bit more in Hoyai, but I'm going to remind you that the scale is 0.1. For FST of sex, we would expect it to at least get up to 0.5 because this is a heterogametic situation here. We expect that to be heterozygous. So far, wash. Huron is worse. We see absolutely nothing signal-wise in Huron between males and females. Again, no strong signals. In superior, we start to see maybe a little bit of something. Artidae has very weak signal. Hoyai, it's getting a little stronger. And then in superior, Kaiai, it's getting there, but it's still not at the level that I would expect. So we're really not thrilled at this point with what we've seen sex-wise in these fish, which is disappointing because we love to be able to genotype non-lethally. So the way in which to be confident about a fish's sex, especially when they're not spawning, is a lethal dissection. And that's not very useful if you want to preserve these fish and raise them in a hatchery setting. So we have done some preliminary attempts at an assay for genotyping that region for sex, and complete bust so far. So to sum that part up for you, there's not much to see, which points us to a couple different things. First of all, Huron having no signal whatsoever is very interesting because at least between Michigan and Superior, the region, very tiny region that we see is consistent. But Huron has nothing at all, which suggests that maybe it has a different sex determining system. And that is very possible. And many lineages of fish, even closely related species, can have completely different sex determining mechanisms. Maybe that region is super tiny and we're having trouble detecting it with this methodology. That might be something that we call polygenic sex determination. So rather than getting a strong signal in one region, because that's where the sex determining system is, there are actually multiple genes across chromosomes that are involved in sex determination that are working kind of together, rather than having a single large effect gene. Yes, it could be very small, like Takafugu. So they used a completely different methodology to determine that sex determiner. And it's a pain. I'd prefer not to do that. But it's potentially what would be necessary. And it's also possible that we have different sex determining mechanisms across these lakes. We just at this point don't really know. And that's something we're going to be working on moving forward. But to kind of overall wrap up what I talked about today is that we're seeing a lot of interesting variation between populations and between species. So I think the subpopulation level stuff going on in Lake Huron is super interesting and has a lot of implications for how we conserve are to die in Lake Huron, whether we need to make sure we're making those their own units to work with. Um, Lots of candidate genes to go ahead and start looking at. It's possible future sampling in Lake Huron of some of the regions we don't have data for yet could reveal further structures. So it's getting complex now. It could be even more complex. It'd be really interesting to try and understand why Lake Huron specifically has this substructuring and we don't see it so much in the other lakes at this point, whether that's something based on the lake itself. Lots of things to consider. And unfortunately, identifying the sex chromosomes in these fish is going to be perhaps harder than we'd hoped, but it's not something we are going to give up on at this point. Just going to pursue some other avenues, I think. But with that, lots of awesome people to thank. Our current version of the genome was assembled by Sarah Chang and Trevor Krabenhoff's lab at University of Buffalo. She saved me a lot of work by doing that. I don't love genome assemblies. But yeah, fire questions at me. If you ask about ecology, I will do my best, but still new to me.
Thank you very much, Kristen. Really, really cool information you shared from the population genetics, and then just the sex determination thing. I assume is new to almost all of us. We haven't really thought about that much with respect to Coregonines before. So the best way to ask questions, if you will, would be to raise your hand and put the camera on to have as best as we can a face-to-face conversation. If that's not possible, you can always write your question in the chat. And yeah, I don't even have time to ask my question. Let's go first, right to Wendy.
Yeah. Hey, great talk. Nice summary of the past work. So, when I worked on this a while ago, one thing I always thought would be a kind of a neat approach to take was to go, because it was always really interesting to me that Stenotis had the marker. So the Inconnu, which you'll be hearing about next week, at one time they were classified in Coregonus and were separated mostly based on life history and morphology, but the genetics is still pretty similar. So I always thought that would be a really great jumping off point for digging into the Carrigines. I don't know if that's come into your consideration for next steps, because that's something where Deano found good markers, and it might be worthwhile to look at, and now that you have more genomic techniques to go back and revisit that, that might be a place that if you can find out where it is in their genome and then do like a comparative genome analysis now that we have this nice new reference genome. I don't know if that's something you've started to consider or would be interested in considering.
I had not, but that would be super cool. There's so many jumping off points, I think, at this point that I think I could go forever and just keep ping-ponging to other groups. There's a lot of like inland lake stuff to think about. And yeah, there's hopefully going to be a couple more reference genomes for some of the other species because Sarah's working on a pan genome project. There's a lot of possibilities there.
Yeah, thanks, Wendy. Maybe one thing we can ask for this webinar, since Kristen's kind of new to the area, is to also tell Kristen your affiliation. So, and of course, you have some background here, which Kristen probably knows anyway, but maybe your affiliation too, Wendy, where you are.
Okay, so I'm with Fisheries and Oceans Canada at the Freshwater Institute in Winnipeg, and I run their genetics program. And I've worked
Cool.
on Coregonid once or twice in my past career.
And how long were you here at the Great Lakes Science Center, Wendy?
20 years.
20 years. There we
Wow.
go. Yeah.
Yeah.
Okay. Thanks so much, Wendy. Caroline.
Hi, I really enjoy your talk. I'm working with Nick Saad at Senior Oswego, and before I played around with a lot of weird sex chromosome stuff. And so I'm just curious if I know Sarah's new genome is great, but I was wondering if you also look, if you see a stronger signal on some scaffold. Just if your region is tiny, then I don't know.
Y-
You might it
Yeah,
might.
we No worries. Yes, we did also look at the unplaced scaffolds, which in her new genome, there's not a lot of them. Did not see any signal out in there either because, yeah, we were concerned, but also those scaffold pieces are 2 megabases or less. So they're really just tiny pieces that didn't find a home. We also are running into the issue of how many chromosomes are there actually, because when they've been karyotyped historically, they say 40. The assembly has 38, 37. Like the numbers are not matching. So there's a lot of stuff underlying that are complexities that come with a rediploidizing tetraploid. So lots of crazy stuff going on.
Thank you, Carolyn. Yeah. Ellen.
Well, hi. See if I can get that to work. Hi. Great talk. Thank you. This was intriguing. I am the University of Vermont retired, a lapsed geneticist. I work on lake trout, so a kindred, but far kindred. And also I've worked on whitefish. So this is a little unfair because you're telling this richness of the ciscoes, right? And anyway, fascinating. Is there any prediction based on what you're finding that that this sort of, as I say, outlier of whitefish could have any of this kind of diversity between rivers and lakes or within in bays, and we have a fragmented lake? Or why is it that they're so very, very different? And as I say, it's a little unfair, but I just wonder if you want to speculate.
That's a
Thanks.
great question. I mean, there's a lot of different opportunities to move into niches to specialize. It might be because these fish are homing to specific locations, and that's kind of causing them not to interbreed anymore and overlap. So they specifically home maybe to Whitney Bay rather than Skim and Cove, and that's as much of a reproductive barrier as they need. I would love to know if it's like a reproductive like behavior driving that, if it's something specific to the environment they're occupying. A lot of different possibilities that it's really exciting to try and pursue.
I wonder if it's just sort of almost capacity of the genome, that flexibility that maybe whitefish don't have. Anyway, fascinating. Thank you.
Mm-hmm.
Thanks, Ellen. Sophia.
I really, really enjoyed your talk, Kristen. Excellent. I have a background in sex chromosomes and omic analyses, but it's so fascinating to see what you've been doing with this system. And I'm a new research associate in the SARD lab at SUNY Oswego. And so I was just curious about the population structure results, and I'm new to the Corgonin system, so I was wondering if you had any thoughts about differences in standing variation in Hoyai and Lake Michigan versus, I guess it was the other lake where it was kind of flipped, where Hoyai had a lot more standing variation than Artidai in Lake Michigan versus, I guess, was it
Lake Huron.
Lake
Yeah.
Huron? Thank you, Bo.
Yeah.
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 So I was just curious if you had, if there were ecological hypotheses, or if you the demographic history that explains that. Yeah.
Yeah, so Amanda and I have talked about this. In Lake Michigan, we know that the Artidai underwent a pretty big bottleneck and a huge population loss. So we think that that might be driving some of that in Lake Michigan. And on the flip side, in Huron, I mean, you could see something similar where you have this huge population loss and it just reduces the diversity overall. It especially because it seems like Hoyai, I believe it's in Huron, they're not very different from each other. So that suggests that they're all intermating. There's constant mixing. So they've not really separated out in any way. And whether or not that's just because they're deep water species and that just makes them more homogeneous, or if it's because there was a huge loss and we had a lot of overfishing and other sorts of things impacting these fish. A lot of possibilities. We are going to be working with historical scale samples, work that's in progress, so we'll be able to actually compare these current populations to what they looked like in the past, and that might help us answer some of these questions, whether or not they were more diverse in the past and they've lost that diversity, or if maybe that's kind of just their holding pattern. So there's a lot of different avenues we're looking at going down to try and address some of these questions.
Very cool. Thanks.
Thank you, Sophie. So Kristen, there were a lot of kind of really cool, surprising results in there that I saw. One, I think it was going back to the population genetics, the variation you found in Cisco and Arteti and Lake Huron. I mean, just Scammon Cove versus Whitney Bay, as you indicated, so close together. I know that people have found, like some of Wendy's work, even with microsatellites, showed some of those differences, or at least showed maybe one of those bays being very different. I can't remember which one. Perry Sound, and then the Riverine. That's the first time I've seen sort of those river samples that Andrew Hansi collected in there, all together on that same plot. Just really cool variation, and to some extent, that I think it's worth mentioning that overlaps or is somewhat consistent with some of the morphological differences that Randy Eschenroder and others have been seeing. So it's nice to see. I mean, I don't know if there's like one-to-one alignment, but morphological differences and now clear genetic differences using the whole, our strongest genomic technique yet. So that's just worth mentioning. But for me, the surprising part of Lake Huron, sorry for all that, was that very little hybridization between Hoyi and Ardii.
Yep.
So you found some evidence of it in Lake Michigan, which I think is also kind of cool and surprising. I think, again, based on some previous work, led in one of Randy Eschenroder's hypothesis, is this hybrid swarm happened more in Lake Huron. You're not seeing evidence of that genetically, at least between those two species, but you did see more evidence of it in Lake Michigan. So have you had much time to sort of reflect on the difference in the Cisco, Hoyi, Arteti hybrids happening more in Michigan than in Huron, at least based on your results?
I think Amanda can probably speak to this better than I can, but based on what I recall of discussions on how these samples were selected, I think we were specifically trying to not include hybrids generally. So some of these individuals have been GT-seeked first. So we were pretty confident in the call for those. So we haven't really done a full evaluation of hybrids in Huron or really anywhere targetedly at this point.
Wow.
Am I right on that, Amanda?
Yeah, so not quite everything, but where we could, we tried to vet things before we sent them for low coverage work with our GT-seq panel. So some of these things were vetted as, okay, we know it's a species, but not everything was. And so I'm not entirely sure that it's a perfect, like, random collection that we see, but it's very interesting to see the differences between Michigan and Huron. And so part of the reflection we might see is that the bloater and Huron are still spawning in deep areas and the Cisco are staying in shallow areas. Without delving too much into stuff, we see a lot of interesting movement of bloater in typically Cisco space in Lake Michigan now. And so perhaps there's more opportunities to hybridize with each other if the location or the depth is starting to overlap more between the one remaining population of Cisco in Lake Michigan and the bloater populations. That is just conjecture. So I don't know. We know that they're occupying a lot more shallow water space during different parts of their lives. But what I do think is interesting is that just morphologically, anything that was a hybrid that was identified in Lake Michigan was always identified as a Ardii. When they went out and field ID'd as bloater, it was always bloater. But sometimes Ardii was a hybrid, not just Ardii.
Yeah, it'd be really cool to see what those, if we have images of those fish, like, would we be shocked that your genetics came back as hybrids on those? I mean, that would be, yeah, very interesting.
Mm.
All right, thanks. Wendy.
Yeah, just to follow up on the hybrid work, it always surprises me a little bit to see so much in Lake Superior, given that when you make them in the lab, they don't survive very well. But it is interesting that I kind of got thinking about this just a little while ago, and it's like, well, whenever we've made the crosses between species, it's been Lake Huron, Lake Michigan, it would be interesting to see if you tried it with Lake Superior fish, would they survive a little bit better? Because, when we've made them in the past using fish from Huron and Michigan, very few survive. So it kind of surprises me why you'd see so many more in the wild. But maybe that's a lake specific thing that's accounting for that, considering the differences in the genetic variation, the standing genetic variation that you're starting to see.
I think of one example that we did, well, not we, Jason Smith and Chris Dye, they were trying to make Kai crosses in Lake Superior, and one of them turned out to be a bloater. So there was the bloater Kai that survived and are still swimming around. Those are now Little Traverse Bay Band hatchery and at our Tunison hatchery. But we don't know if they are, if they can back cross or if those F1s but that's one example in Lake Superior of that successful cross to produce the F1s that have been seen in like some of the RADseq or the GTC data as well. Yeah, Amanda.
Yeah, I was just going to follow up on Wendy's comment and say, like, it's really an unknown area, because it's not uncommon for things to hybridize to F1 like all over the place and even in very distantly related species. But knowing how and in what direction and the percentage that actually does survive, like I think what he said, we do have evidence, both in just looking at our genetics and then some of the work that she's done, that there's not like it's not like necessarily beneficial to hybridize, that it's a beneficial state or anything. And that it might actually, there may be incompatibilities in there. But another thing I was just going to say while we've got folks on that have done chromosome or genome work, if anybody's got good connections to karyotyping, that's sort of one of the next steps that we're going to try and be going into, because there's work out of Cornell and there's work out of the University of Wisconsin-Madison over the past probably 60 years where they karyotyped Great Lakes ciscoes. And like Kristen said, 40, sometimes 41 chromosomes is what they would call. And so far, we've only been able to resolve, I see Trevor shaking his head, resolve 38. And the other thing is that there's been some work in Russia karyotyping, and they talk about seeing B chromosomes, which are these little tiny fragments of chromosomes that hang out and can be passed on as well. And there's no discussion of B chromosomes. There's no images showing B chromosomes. So we don't know if there are B chromosomes as well that might be carrying important genetic material for possibly for sex determination even. So it's another extra thing. So I have reached out preliminary to some folks in Texas that do it in more of an agricultural manner. But if anybody has any connections to folks that karyotype, do let us know, because that's one of the next steps, is trying to converge what we see in terms of the genome resources and getting some like more recent imaging of the chromosomal fragments that we see.
Thanks, Amanda. And Wendy put in the chat that the Russians are still karyotyping. So, Trevor.
Yeah, great talk, Kristen. Trevor Krabnoff, University at Buffalo. I really enjoyed that. I had a question. So regarding sex determination, I think you sold it a little bit short. I think the chromosome 31 looks really promising. I don't know if that's the same region Amanda found before, but
Yeah, it is. For
okay.
me, no, go ahead. Finish your question and then I'll.
So we looked at some of the genes in that region before when Amanda initially saw something there, and there's a lot of I think promising genes in there maybe it's just a matter of winnowing that data down a little bit. But the other question, so I'm not sure if I'm remembering correctly. I think we sequenced a female in the genome assembly, and maybe you said this, but if it's male specific region, it might just not be there. So I wonder if there's some reads that show up in the male samples that are not present in the female genome. So maybe just a read mapping kind of approach. I don't know if you've tried any structural variant analysis or anything.
I haven't looked at structural variant analysis yet. I would expect that those variants would map, or those reads that are pertaining to the male would map elsewhere against the female. The problem with having what is presumably a pretty small, not super diverged region is you can't really use a read mapping or read depth approach to detecting sex chromosomes. You're just not going to get enough difference. Like my background is in cichlids, very similar situation where most of the time the chromosomes are homomorphic, and we've never had success with read mapping. So yeah, there's a couple different rabbit holes we're going to have to go down to try and target that region. The reason I'm not super thrilled with the strength of the signal on 31 is it's just not clearing thresholds that I normally use for distinguishing a sex determining region. Like you would really expect that FST to be between 0.5 and 0.7. And usually in my previous pipeline, it was built to filter on that. I'm using a kind of a different setup here, so we're going to see that signal, but it's really, it gets to about 0.4, which is, it's just borderline, and it's only borderline in Chi-I, and it's just not, it doesn't have me convinced, essentially. I don't know. I wasn't thrilled when those results came out. I was like, I was really hoping for just a strong, strong signal, and then I could go dig around and look for candidate genes. And when I look for candidate genes, there are obviously what we call the usual suspects in fish: GSDF, DMRT1, AMR, or yes, AMH. But I'm also kind of at a point where my previous experience is that yes, there are certain pathways where you can have a sex determining gene continuously rise out of, but the Salmonid gene is a complete dark horse. So you risk a level of bias when you go in and that sort of thing. So lots of things to look at gene wise in that region, because we're going to look anyway, even if I'm not, thrilled.
Thank you.
Thanks, Trevor. Sophie.
Oh yeah, just a tiny, tiny question to follow up on that, Kristen. I was wondering what the sliding window size was for your FSD analysis, or if you tried different windows.
Yeah, it's 25,000 base pairs. I've looked at these plots also, just the raw FST, and it bumps Chi up just a little bit, but it's still not it's still not meeting that threshold, which is driving me crazy. Yeah.
Okay.
And 25,000 I don't feel is excessive. I mean, I can re-window stuff. I just don't think it's gonna take that signal where I want it to be.
Yeah. Very fascinating.
Caroline.
Hi, it's just to follow up again, especially with Trevor comment. I was wondering if you were gonna try something timer-based approach to get rid of like the reference
Yeah.
Trevor said.
The problem is we're working with low coverage data, and I'm not confident that we'd get really great K-mers out of that. It's an approach I'm familiar with, and it's definitely something I could try. It's just I worry about us missing out on a lot of data that we kind of get back by using a genotype likelihood approach. But yeah, it's definitely something to consider. It won't help any of the Ardidai to Ardidai comparisons, but with the Chi-I, maybe. It's possible that the Chi-I region is not present in the Ardidai genome. So yeah, that's an approach that I can possibly take, is to try and remove some of that reference bias. So many different possibilities.
And you also show the SDY gel pictures for Guinness, and I can't remember if they ever published a sequence that they got from the male and the female. If you look at the sequence, just because of the weird thing that's happening also with Shynock and also salmon, that you have like a weird autosomal X-linked copy that's present, but there's like a SNP difference between the two, and I can't remember like the sequence basically for
Yeah, I don't remember off the top of my head, but I'll have to go back and double check and see.
Well, they do have the gene region available on NCBI, and I did a quick map to an earlier version of the genome out of Trevor's lab, which I'm sure it hasn't changed radically, but I believe it came out on chromosome 25, where we saw absolutely no, not even like a tiny peak, just nothing, just flat between males and females by species.
Thank you.
Mm-hmm.
I might ask one more question. That's a simple one, I hope. For Lake Superior, for the Arteti, you talked about the alternate population, and then like you had just called it the Apostle Island, or the more broad one. Have you ruled out that that alternate population is not hybrids? I mean, when you see that structure plot, it kind of looks like hybridization may be going on, but
Amanda's over there shaking her head. No, I think it pretty clearly on the GTC also comes out as Artidai, and we would see it kind of pulling that way on the PCA. I would presume that it would be either hybridized with Hoyi or whitefish, and it's not behaving like that. It's very clearly part of the Artidai cluster. It's just basically makes two subclusters. So they're Artidai, they're just weird Artidai.
Yeah, it's very interesting. I don't know, Kristen, if you could back up to your PCA of Lake Superior. These are very different, but they're not hybrids. They're definitely a very different population of cisco. So if you look at the red cluster, it's the little tail end of that red cluster off, like on the most left, upper left. There's a little cluster that's up and away from the rest of the cisco. So there's like the standard genotype that we see in Superior. You can see what looks like possible hybrids sort of trailing towards bloater and bloater trailing up towards that cisco. And then that little cloud up off is this weird population. And it's a real mystery right now because we sometimes call it Grand Portage because the first time we detected it was in some work with Dan Yule out of Grand Portage Bay, where there were some larvae we identified and a couple of adults we identified with rad sequencing. But it wasn't until we started using GTseq really broadly across a lot of the work that the Lake Superior Biological Station is doing, IDing larvae across the entire lake, that we started getting these messed up identification, like clustering for our IDs. And it was this weird population throwing in extra variants. And so we see larvae from this population of unknown origin all along the northern part of Lake Superior, sort of like Nipigon Bay area, and that area. It comes out of it. And so I have no idea where this comes from.
yeah,
It seems to integress readily with the standard form. Most of the individuals that we used for this analysis, just to compare the differences, are larval samples, if not all of them, I think. And so my current running theory is there's some kind of lake population that might have an outflow, but the inability of an inflow from the lake. But it's a mystery.
It was the structure plot that made me ask the question, but is it just that the artifact of forcing structure to make those colors, is that the result? Not suggesting that it's that's mostly Hoyi in there?
The weird ones actually, I believe, are the standard Artidai. And the solid red ones, I believe, are the Grand Portage, the alternate strain. Yeah, so it's calling essentially our normal Artidai something in between bloater because of how similar like the genotypes are there, like if it's putting in there. But yeah, my best guess, I did a little bit of analysis with some RadSeq data I have to see if it was maybe coming from like Lake Nipigon or something like that. And it's not a Western lineage. So it's not coming from like the Pigeon River. It's not coming from something that could have spilled out into the lake from Lake Nipigon. It's the same lineage of cisco that we see in the Great Lakes, which is more eastern or sort of an integressed version. So yeah, my best guess is somewhere along that northern shoreline of Ontario is some kind of embayment where we have
Connected lake. Yeah.
river spawning or a lake that's connected where they can outflow larvae into the big lake, but it's not easy or common for adults to get back up in there.
And Andrew Hansi has in the chat, you addressed part of that, but by looking at Nipigon, of course, Black and Thunder are very more connected to Lake Superior. Maybe even some of your samples are from Thunder Bay and Black Bay. I know that Daniel has spawning. He's done spawning studies in there. I don't know if he's actually collected fish for genetics that are. Yeah. So it looks like you do have samples in those embayments.
I don't know if it's the Artidae though.
Oh, that's true.
Yeah, it's
we
hard to
may not
tell.
have with RAD-seq data. What's interesting is with RAD-seq data, with samples from Thunder Bay, a lot of samples from Thunder Bay, a lot of it samples from Black Bay. I don't think I might have had samples from Nipigon Bay. Those looked very similar to what we found in the Apostle Islands. And when I had adult collections of what turns out to be this alternate population, they were coming from along like the western arm, along like Minnesota there. So maybe some stuff near Grand Portage or like Minnesota waters of Lake Superior. And it wasn't much. It was maybe 7. And I actually have old PCA plots from RAD-seq data where that streaks out of the main Cisco plot, those individuals. But once we started getting the larval data from the Lake Superior Biological Station, you could see this like swath of stuff.
Much more distributed. Yeah.
Yeah.
All along that northern shoreline.
It seemed really intense up there in the northern part of Superior. And because of the way the currents flow, my best guess is maybe the currents are washing the larvae and they're just settling in that area as adults, or recruiting to that area as adults. And it seemed they readily integress with the population or genotype that we see elsewhere in the lake.
Okay, Wendy, Kristen, I don't know if you could read Wendy's chat there, suggesting another way of looking at some of the high dimensional data.
Yeah, I'm familiar with UMass. I've not actually seen it used in this context. I'm more familiar with it for like single cell mapping, but yeah, it's another consideration to try and pull things out.
Okay. Well, this has been a great discussion. Thank you all so much for all the great questions. I think we'll call it there. If you can sort of send Kristen a thanks in the chat or do the hand thing. There we go, to make this a little bit more personable. That's great. Thanks again for your talk, and we hope to see many of you again next week to learn more about, I will be learning more about In Canoe from Randy Brown. So thanks again. Have a great weekend, everyone.